my background is soft condensed matter physics. I am not a doctor. I'm not trained as a doctor. I'm not really trained in medical practice at all, actually. But one of the factors that, while going through grad school, I come from India, I grew up in India. One of the factors that at least, when you get trained as an applied physicist, you really think about making stuff. And there is a large amount of research that we do, which I'll not talk about today at all. Maybe I'll just briefly mention it, which trains us to think about making things. And this is really a path that grew out of that. And now my group essentially has 50% of the projects are really in global health and this idea of frugal science. And I kind of attach this word science to frugality because I actually believe there is a lot of undiscovered science to begin with in spaces like this. So I'm going to try to quantify and say, why do we really call it frugal science to begin with? Okay, and since I didn't actually prepare talks, so I might go back and forth between slides to remember what I want to say. So the biggest thing about thinking about, for me, in something like this is when you start making stuff, you kind of consider yourself as a technologist, and many of you, depending on if there is an interest in an engineering school, might classify yourself as technologist. And I think one of the aspects that ends up happening is, especially for global health, there is a huge interest and, of course, demand to thinking about the intersection of new technologies with global health. And the challenge that comes in is it context specific. And that's really actually a hard challenge into thinking about pushing technology into a problem just for the sake because we can do it is actually something that bothers me quite a lot. So there are a large number of things, and the reason this talk was kind of like a last-minute thing is in a week from now, we're leaving for Uganda and Nigeria and frantically trying to make as many instruments that we can, is really this context of what can we know early on before taking ideas out in the field on a larger scale. And scale is something that's also important that I definitely want to talk. So if I forget about scale, please remind me, because I think technologies that scale is an important aspect of thinking about solving problems. If the goal here, to being honest, is actually trying to solve problems. So this is my group. I always forget the credits. So this is really actually one aspect of what is really fun to being at Stanford is just really being able to work with many of these students. We were at a maker fair, which if some of you are in San Francisco and around this area, you often go to. And the way I think about the lab, let me just give you a context of the types of areas that we work in. I call it a circus primarily because of this idea of, we're trying to have fun, and you will see that in some of the projects. But the other side of the circus is really how areas intersect. And in the past, before running my own group, I was just a graduate student or a postdoc just working very focused on one problem. I hadn't realized this, that the synergy of these teams, so one half of the team really what we work on are extremely primitive organisms, so multicellular organisms that are really at the root of multicellular life. Sponges is something that you've all seen if you go snorkeling and things like that. These things are, in my mind, and it's a hot debated thing that we're trying to fight, these things are even simpler than sponges and are probably the simplest animals known on the planet. And why we think about that is primarily just because we know nothing about them. We don't know where they are. We don't know anything from a perspective of where they originated, what actually led to other metazoans from something like that. And it's just a big question. And we go out at Hopkins, capture these things, and then try to figure out how they work. So this is one aspect of organismic biophysics that we work on. And then the second aspect of the group is really focused on tools and technologies that are applied to global health. There are a couple of problems that we work on that are not directly global health oriented. But this circus and the fact that a lot of imaging stuff that actually came out that I'll talk about was initially thought for other problems that we work on is actually a fantastic thing personally for me as a young lab. We've been at Stanford around for 2 years now. So the other aspect, I was very happy to actually, somebody mentioned there is a pathologist in the room, which is absolutely great. And also this is from a context of thinking about people who really think about these problems on a daily basis as their career as well. So I just want to get a sense for what type of people do we have in this room. How many of you are inventors and, in a very broad sense? And tinkerers is actually a better word probably. And then what class of people are business-oriented and think about taking stuff out and applying a business mindset to it. And then, the question reason I put this is, as an academic, we often think about coming up with a solution, and then we pass the buck to some of you guys. Because, somebody was mentioning, so how many people are using this thing? And, as of today, we are doing all kinds of things in manufacturing. We're in a manufacturing phase right now. But specifically with a project like this, I think it might be really interesting to have a little bit of conversations based around, when you come up with something like this, which are the directions that the business models actually go. And I'll throw out to you what we are thinking about, but that would be a fun conversation that would actually be useful to me personally. So here is a logo of American Society of Tropical Medicine and Hygiene, and you see two things there. There is a microscope and a mosquito. And one of the things that I was surprised with is how long has this thing been identified? So from a context of the society started in 1903, and it is one of the largest societies for tropical medicine, and it's, in a couple of weeks, actually, the meeting is in DC that we will go and present some of this stuff to. And the thing that I feel sometimes is I find it absolutely amazing what we can do in a given location from a technological perspective, and still you can take a short flight and find yourself in situations where there are absolutely no solutions. And so the fact that something like that can exist both motivates me, but at the same time, I kind of find it as a paradox, because I do believe that we've kind of patted ourselves on the back a little too much in thinking about how much progress that we have made, because many a time some of these factors have not penetrated at all in the global context. And one fun way of describing what I just said to you is I sometimes, all of you, receive junk mail, and this is an ad that I got in my mail, which says, if you live here, you can have high-speed internet, and on the bottom it says, high-speed internet for all mankind. And what bothers me is about a bubble that we ourselves create. This is not all mankind. And it's a joke, but it's actually also a reality in some sense, and that's a reality that we have to deal with as technologists in some way. Okay, so here is the only sad slide of the talk, and then we'll probably not talk about problems, but really talk about solutions. So choose any of these numbers, and this is sort of preaching to the choir in some sense. You all have, at some point of time, based on travel and things like that, seen the other side of the equation. And if you choose and read any of these things, it's very depressing. There are a few things that are good. And I think there is a turn of the tide in thinking about we're at least using elimination words for many of these diseases nowadays. So infectious diseases is something that's one of the biggest burdens. But the challenge comes in is when you have such large numbers to deal with, individual efforts completely get thwarted out. They do not scale. So that's one thing that I'll probably actually mention and talk about a couple times. Okay, so here is the fun start of thinking about demos. So I had brought some tape with me, and now I cannot find it. Oh, perfect. So why do I call it frugal science, and why do we think about this from a context of science? So you all know X-rays, right? X-rays, you get a trouble, you get a broken bone, you want to get it seen. X-rays don't exist at majority of places in developing countries' context. And 3 years ago, I was reading a paper. This is a cover of Nature, and it turns out, some of you know this, is if you pull, and this actually works with Scotch tape, but nobody's really tested a large number of other tapes, is the sound that you hear when I'm going to pull this. That sound just actually produced X-rays. And, it sort of messes with your head sometimes when you're thinking, I know physics. How the hell does something like that, as a simple phenomena like tape peeling, actually produce X-rays? And this is actually not the first time people discovered it. It was a Russian guy in the 1950s, and even before that, a couple of people, when they had just started coming with sensitive X-ray detectors, they claimed that, yes, X-rays could be produced. We keep a lot of literature to the side. Nobody looks at it, and then suddenly a physicist at UCLA essentially looked at it from a context. And the simplest thing to understand it is there are phenomena when you peel. So if you were to zoom in and see really at the edge of what I'm peeling right there, that peeling is essentially coming from a large number of bonds that were formed before that you're suddenly breaking. And nobody really has a good explanation for why such high energy ionization can actually be produced, but you can literally take an X-ray of a finger with something like that as a source. And it's not there yet. It's not actually a commercial product. It does require to put the Scotch tape a little bit in vacuum because the X-rays are pretty weak. But at the same time, that image that you're looking at is literally an X-ray. All it is primarily the source is Scotch tape and being pulled by a motor. And so this is what I mean by frugal science, is the idea that many times when you do make discoveries in science that are very orthogonal to what we have thought about, it completely changes. With something like this, if this as an X-ray source actually turns out to be a powerful source enough, you could think of conformal X-rays. All kinds of things completely change in how the infrastructure of the business models of how X-ray imaging should be delivered. But the challenge here is how do you find these hidden nuggets, and how do you get people to actually agree that frugality is actually both intellectually interesting. It's not just about off-the-shelf parts, and also there is deep science and then actually making the next generation physicists be interested in phenomena around you that you just think are mundane. And at that same time, not everybody wants to be a particle physicist, but at the same time, there is that equation of the science of mundane was done in the 1900s and the 1800s, and now that's become like a niche. But one of the aspects that comes out with something like this is there are large number of phenomena that we actually don't even understand. And then this other applied aspect of the beauty of this work that I bring it up all the time is somebody was thinking from the context of not just, yes, this emits X-rays, but what can it actually do? So they are actually in test trials. They have a new source that they're testing. I don't know where the commercialization side of it is, but at the same time, they're trying to commercialize this, but we still have very little of why it actually works, which is a fascinating question to begin with. Okay, so I'm going to switch. And is there a clock somewhere here so I know how much time? Okay, so I'm going to go a little bit faster. Let me tell you about just one thing that is of interest to me before I jump and only then talk about microscopes. And this is the only quiz in this talk, even though it's titled No Quizzes. So there are two other areas that my lab works in. One is from a context of making microscopy really available to everybody, but the other aspect is not on the human side, but on the vector side, which is mosquitoes. So this is the quiz. How many species of mosquitoes do you guys think are on this planet? Okay, if you obviously know the answer, okay, we'll start with, okay, tell me. How many rough number of species? Go for it. 20, okay. We're going to go. 196, okay, yes. 2,000, yes. 150, okay, I want more answers. 5,000, yes. Two, okay.
We're talking genuses, I think,
3, but I meant species, okay. 20,000, okay, so that's the number. So think about from a context of, and this is actually a fascinating problem. This is one vector that's troubled us. If there was one thing in human history, if you read, there's a great book out called Fever. If you just read that book, it tells you how historic events were actually shaped by this one vector. There was somebody who said 2,000, so I'm going to give it to you. It's around 3,495, but again, with insects, as you all know, there is so much biodiversity that this count keeps changing. And we do discover new things. You read probably in the news last week, they discovered a dengue vector in Menlo Park. Did you guys read that? Yeah, so it turns out they just found a dengue, and it doesn't mean that there is dengue, which is a disease that's caused by this very specific vector. And also, if you all go hiking, there's plenty of malaria-carrying vectors up in Yosemite and most national parks, especially in California. So the fascinating thing about this is on the human side, we have focused, but at the same time, many of these diseases also have an ecological context to it. So one technological space that we are working on is trying to understand. So when you go out there, the current definition of how do you really understand what vectors and diseases are out there is a technology which is called a human bait. So if I go out, I'm leaving for Uganda in 2 weeks. When I go out there, if I take one of these pipes and I sit with my hands open like this, and I wait for something to bite me, the moment that mosquito is biting me, I'm going to pluck it with a thing, and I have it captured. Typically, so this is how you think about which species of mosquitoes are biting on humans at a given place. And also, I would like to know whether that place has a specific infection or not. So for example, for Uganda, if there is a ton of malaria, and typically for a disease like malaria, the number of mosquitoes that are actually carrying the disease that are active even in endemic areas is of the order of 0.01, 0.001. So you've got to catch a ton of mosquitoes. So tens of thousands of mosquitoes are collected, and this is from my experience in Thailand where I was sitting, and I saw 100 microscopists sitting in a room. They were doing a survey for Thailand-Burma border, which is the deadliest place for this disease because they're actually starting to see a ton of resistance around there. And typically what ends up happening is people collect these mosquitoes, you sit on a microscope and you dissect it. So if I was to ask you to take the head of a mosquito and then find its salivary gland, which is somewhere of the order of 50 to 100 microns, and in there look for an active sporozoite. And this is actually how malaria was discovered in mosquitoes. And do that for tens of thousands of insects, that's how we claim that there is a place that actually has a disease. And so from a technologist perspective, you go, wow, all the maps that we've been seeing that you actually see that say that this species is here, this species is here, and this is the disease that exists, a lot of it is with humans, but when you go into the vectors, if you're trying to do vector control, most of that is mathematical modeling because we have no way of actually going out there and talking to the vectors. So the thing that we started working in that space is thinking about what could we do, and an accident happened in the lab. So this is work of a graduate student, Hari Priya. We have active colonies. It's the only mosquito colony on campus, and we're very proud of that. If you come to the lab, it's double containment. This is mostly non-infectious, but we are getting into infected insects at CDC. She did a very simple thing. We were trying to keep these alive, and they were not feeding anything. And we all know, mosquitoes don't drink blood because they want it for nourishment. They drink sugar. Sugar water and raisins is what they want. This is really for producing eggs. But they were not drinking the sugar we were providing them. So one of the undergraduates in the lab went to the store and got Jell-O. And they thought, got plenty of sugar, let's feed them Jell-O. And they put these little Jell-O pockets, and they absolutely love it. And the accident that happened was inside that Jell-O, before throwing that thing away, accidentally this person actually looked under the microscope. And what she found was every single bite that the mosquito was making, she found salivary droplets. So now you see where I'm going with this, is the idea that without doing anything, rather than catching mosquitoes, you could actually be doing analysis of individual. So at this point, if you were to ask me, for a given species, it's not even known how much saliva is injected in you. So if you would like to know the probability of whether you will get infected 100%, those numbers are not known because we can't really do the single droplet analysis. And the other half of my lab works on droplet fluidics to really thinking about now if I could collect tens of thousands of these droplets, we essentially have an assay. And the other accident that Hari discovered was, it turns out there is enough DNA in that saliva from the mosquito, not just the parasite, that we can detect just using multiplex PCR, both the vector and the species that it's carrying. And suddenly, just like if you've read microarrays, immediately you get this picture of what is where and what diseases are they carrying. Yes?
This is just a practical question. Will they prefer a bowl of Jell-O at a picnic to us?
No. We are really perfect for them. And the reason I actually say that is primarily we are doing a bait screen with this. So now, let me tell you what the product looks like, because most of these ideas and projects, we only work on them because we want to make products. This is an academic place, but one of the biggest aspects of working in global health is making products. Just like software people just make products. So the product essentially looks like this sheet that essentially has tiny, tiny, tiny pockets of hydrogel or oil, and you paste it, hopefully in a couple of years, the goal is we would be distributing these. You paste it outside your home for a little bit. It has odorants that are specifically designed for given species and also designed for females, because that's what you're really targeting. Females are the ones that are biting humans. And all you do is we bring them back into the lab, and then we run our assays on this in a central place. So this is our sort of goal of thinking about ultra-large-scale surveillance. And one of the things that I find surveillance fascinating is primarily because of this context of, we've done a significant amount of stuff on humans, but the vectors is just still very trivial technologies. The technologies that really exist on the vector side are people go and collect stuff. So I wanted to give this as a context just because when we have questions and things like this, this is something that would be fun to actually talk about. Okay, so I'm going to switch now. Let's talk about microscopes. Yes? Oh, what I mean by vector is an organism that primarily carries the disease. Many parasites have life cycles that are in two hosts. We are one of the hosts for malaria, but the other host is a mosquito. So the vector primarily is a guy who carries this and brings it to another human, for example, in our scenario. So vector, just like it's a transporter, essentially. And if you all like drone analogies, there are drones flying around that we should be really worried about all the time, and we have no control on those. And so that's really one of the aspects that you have to think about. Okay, I'm going to stop scaring you. There are very few things that are out here, so you shouldn't really be worried too much about it immediately. But actually, last summer was the highest West Nile infection in the United States. It's the peak. And when you look at the data that CDC gets and the places for mosquitoes and places where there were actually humans that got infected, there's no correlation. So we're not sampling at all because we don't have the government. Also, we have a government now, but no money to actually support any surveillance, for example. It is a hard challenge. Okay, let's switch and talk about what I promised you, and interrupt me as much as we can, and we're going to try to do some demos instead of slides. So one of the things that I want to show you, though, because I enjoy history of science, so this image is actually the very first moment. So microscopes are interesting, because microscopes have shaped societies in very interesting ways. This is an image as a joke from the Wellcome Society in UK of a British mom drinking her tea and freaking out that there are monsters in her cup of tea, this is Thames River. And it turned out this was the moment when microscopes were not just research, but you could just go to a place and buy it. And suddenly people started realizing there was this complete panic reaction that everywhere around you there are infectious things and there are monsters. And you can see some of these things look like monsters. But I'll show you some parasites actually do look like monsters. And I have a ton of parasites up in the top in front that at the very end of the session we're going to look at those. So this is one aspect of the equation that if microscopy became available in developed countries, it actually changed people's perception. If you have a hard time convincing your kids to wash hands, have them look under a microscope. This is one of the hardest problems in sanitation is kids don't wash their hands. You go to the bathroom, it's obviously built in that you have to wash your hands. Go out anywhere in the world, this is one of the hardest problems for diarrheal diseases. And that's primarily is the perception. How many of you have actually seen a bacteria? Like the hand looks clean. There was a question that I missed. No, right? Okay, so this is my other favorite image from a history of science perspective. Does anybody know who that is? Gandhi, right? But there's a paradox in this image. What is the paradox? He's actually using a British microscope. And for people who know a little bit of Gandhi's philosophy, this was around the non-cooperation movement. He's not wearing pants. He was just wearing a piece of cloth because he was burning British clothes. But at the same time, he knew the value because he spent a lot of time on leprosy. And so at his place, there was leprosy diagnosis, and he was very interested in that disease per se. And so this is Gandhis Microscope, and it turns out, I'm going to show you one more image. This is Ronald Ross. This is the person who looked into a mosquito, again in India, who for the very first time confirmed that malarial parasite is actually transmitted through mosquitoes. And he has a fascinating history of how many times he actually gave up on this idea before he finally actually concurred that, yes, this happens. And you notice the microscope right there. And since we are on that history of microscopes, I bought something for you that some of you might enjoy seeing. This is now a roll from the 1850s all the way till present days. And some of these are pieces of art. And one of my favorites is going to come in a few seconds. They're craft right there. And this is a craft primarily because, as manufacturers, we really take pride in what we build. And microscopes kept getting more and more and more complex, and they turned into research instruments. And if I had a whiteboard, at this point, I wanted to draw a curve. But imagine if I'm drawing this curve. This axis is performance, and this axis is cost. And so essentially what we ended up being was in the right corner. Fantastic performance, diffraction limited, and now everybody talks about super resolution microscopy, where you can see below the wavelength of light. When light at that point is fuzzy, you can actually still see an object. So fascinating. And then when you look at the cost, my wife is also a biologist at UCSF. She's a faculty there. She just bought a $500,000 microscope. And that's the cost axis. And for what it needs to do, fundamentally, it's actually extremely important. But now, when you look at this axis and start thinking about what could you do from a context of if microscopy is important for society, and I'm going to show you in one slide why that is, you really have to think about this performance and cost axis. That's everything. And that's really sort of stepping off of our pedestal of saying, OK, what is the best we can do, but what is the best we can do at that cost that it could some scale? And that was really the intuition about the project that I'm going to tell you about. OK, so many diseases have faces, and somebody already mentioned TB for a second, but I want to bring and show you a couple of other diseases that you might have never actually seen before or heard of. One of the fascinating things about microscopy is it's still the gold standard for many diseases. And it's debated because with malaria, microscopy did not penetrate, and so RDTs have penetrated, but it costs $1 or $2, and it's not sustainable from a context of being able to do the diagnosis. And the context really comes from, it's not completely trivial, but the standards for building microscope slides have percolated everywhere. But one of the biggest questions, and this is something that we experienced in some of our work in Thailand, was there were microscopes. Zeiss has a contract with WHO. They ship all kinds of instruments that sit around there. But one of the things that ends up happening is the microscope is so expensive, it's 5 times the annual salary of the guy who has the key to that room and whose person's responsibility it is. And so most of the scenarios, and when you think about how do you become a good microscopist, is by spending time on a microscope. And most of the time, people are scared of even touching this 100x objective out in field setting. In hospitals, they're more comfortable. They have more influx of money. So most of the things that we focus and think about is what could you do truly in field settings. So the thing that we came up with, and this is sort of one context of the inspiration, and then I'm just going to show you some actual demos, is I was visiting a matchstick factory at a point of time, and I was baffled with the throughput of how many matchsticks. Can anybody guess how many matchsticks a plant can make in, say, a month or, yeah? 5,000, okay. Yeah. 5 billion, that's actually close. Yeah, in billions. And that's primarily because it's a roll-to-roll process. Trees go in and packaged matchsticks come out. So really have to think about manufacturing. So we kind of stumbled upon this idea that if we want to solve things, we got to have high-performance instruments that are good enough for diseases. We're going to make instruments that are specific to diseases, so not all bells and whistles in a given instrument, but that are very targeted to a given disease. And the third thing that we thought about, that it has to be manufactured in a flat manner. So the whole idea of what we call a Foldscope or an origami microscope. And so this is an invention that there are 2 students. There's Jim, who's actually in the room, and then another student who is now has a job somewhere. And I'm going to show you some parts. So in this envelope, I have somewhere around 10, 15 microscopes. And most of the fun things that you can do is you don't really have to worry about care because they're built and designed for areas that we, and this is actually some of the leftovers from Jim's last trip to India. So many of them are beat up, but I want to give you some context of what an instrument actually looks like. So I'm going to take some of these out. So this, for example, is one of these instruments. It has the light source, all the optics is printed. There's a little place that you put glass slides, so this just happens to be, we're not changing this interface because this interface is actually important and has been optimized for many different things. But this in itself is completely self-sufficient for specific types of diseases. So I'm going to do a couple of demos now to give you a context of what this looks like. The matchstick factory connection is actually large-scale manufacturing of optics and flat manufacturing. So this is a folded instrument, but what it actually looks like when it starts is, and these are some, we have very big interest in education. We're rolling out 10,000 instruments that we're going to give out to kids around in the United States. So this is some of the prototypes of what some of these things will look like. You would get a sheet. So the matchstick analogy primarily is the manufacturing aspects of it is completely planar. You have an optical stage, you have a sample stage, and then you essentially have a light stage. And all of them come together later at a point when it's folded. And the origami context is, it turns out you need alignment for optics, right? It makes sense. And for most optical instruments, the fact that your objective costs $10,000 is somebody sitting and aligning. And there are lots of machines that are involved in optical alignment, which is expensive. Origami is interesting because, and this was another accident that Jim stumbled upon, is when we fold things, we can get for a paper, this paper is somewhere of the order of 150, 200 microns. You can get an optical alignment just primarily by folding that's of the order of 10 microns. So that's phenomenal for an object like paper, and that comes really from the methods of folding, is the fact that folds have constraints. So whenever you make a swan, you really have to ask yourself a question of how close is this object to the mathematical object of a swan that you were going to fold. And if you follow some of the rules that we kind of came up with in this origami context, you can get to tens of microns by human folding. So this was another reason why it's manufactured this way. We're really going to run out of time, so let's actually do demos and hit the lights. So one of the contexts that came up was, how do you really actually teach people? So I'm sort of in the camp that you have to have human infrastructure. Technological infrastructure you can build. Human infrastructure is not existent. How many pathologists are there in Ethiopia? No, very few. The place we're going in Nigeria is a training camp for microscopists, and they train all the microscopists that are in that region of West Africa, and ended up being a context that that's really what you want to also invest in. So I'm of the camp that sort of, one day we will have Watson that's going to know everything, but I want to solve the problem in the next 5 years context, and that really implies we have to invest in training. So while thinking about training, Jim came up with this idea of what we call projection microscopes. So we're going to hit the light. It's almost the exact same microscope, except it's also built as a projector. And I typically do these demos in my class. Before it gets too dark, I'm going to try to find, okay. So this is a low mag objective, and I'm not sure if it's going to work. We just charged the batteries in some sense. I just put in a slide there. It's almost exactly the same, except it has a light source that's a little bit brighter. So tell me if you can actually see anything on the substrate here. Do all of you at the back see something? Okay, so and if not, I'm going to go close, which changes intensity. So you see something blurry there, and I'm going to now try focusing. So what you're looking at right there, tell me if anybody's figured out what that is. Can anybody know what that thing is? Okay, another. Yes, it is a mosquito. You can find that from its antennas. And the thing that you're looking at is, this is the head, that's the eye, there are the antennas. I'm going to focus on tarses. I absolutely love tarses. This is really what pokes you right there. Do you see that? This is really what gets inserted in your hand, and then most of the time there is another tube right in there if you look. The one that's going below it, there are 2 tubes there. The second one is really what actually sucks blood. And so now since I have this, can you hit the light one more second? So I had a friend who just gave me a ton of slides that he came back from a field trip in Uganda. And I want to show you some things from there. And then I think we're going to run out of time. So people who are excited, we can come back down here and look at a whole bunch of things. There are some diseases in there that even I haven't ever looked. So this is a fun one that I found. This is called Loa Loa. And have any of you heard of Loa Loa? OK, that's good. This is essentially a filarial disease. And one of the things that happens is you should just think of many of these things as worms wriggling around in your body. But one of the challenges is that there are many co-infections. There are drugs for this. But if you have 2 filarial diseases, you might actually be in a position where the drug, mass drug administration, is one of the biggest ways you can eliminate diseases. If nobody had a disease in a human population, the vector can keep biting. Eventually, the disease will die. But the challenge is that there are certain diseases that if you have a co-infection, mass drug administration is going to kill you because it produces toxins. And so, of course, no government, even if 500 people die because of mass drug administration, that's actually a pretty terrible thing. So this is one of those things. So let's actually try to find, and I was just looking at my office a few minutes ago before coming here. So I'm going to see whether we find it. That also gives you a context of thinking about what are we talking about. If I land somewhere, all I would have to do for the slide that I'm about to show you is prick a finger, make a drop on a glass slide. Then you make a thin smear, which is you basically take the slide and you shear it to form a single monolayer of cells. And this is stained with Giemsa, which involves a dye that's available everywhere. You put this thing, depending on people, from 30 minutes to an hour. And then you take it out and you look at it. So if we don't see Loa Loa, we'll first also see blood samples. So right there, each one of those little red dots that you see that are connected together are actually blood cells. So now I'm going to navigate around since I don't know. So you see those little blue dots? That, it turns out, is something that's stained with Giemsa. And then you will look at the shape and you realize that's not the filarial worm. So I'm going to keep looking. And this is kind of a fun context because I don't know where this thing is. OK, right there. How many of you can see a long thread-like blue object? I'm going to magnify it. So magnification is just that. And if we were in a dark room, you would see that little blue dot. OK, so I'm going to come close now. Watch the right at the center. Am I blocking the image? Yeah. I see. OK, so I'm going to make a tangential right there. What do you guys see right at the center there? So there is a blue dot, but also it's like a little thread. So what we're going to do is I'll pass this to Jim. And he can pass it and show it to some of these people. But just for the front row right here. All right, so while you guys keep looking, let me continue what else I wanted to mention. Yes, so that's a very big point. So we didn't talk about cost at all. And I'm just going to show this video while this is what the instrument looks like. This is one of the videos, and I hope there is no sound there. OK, so from a perspective of manufacturing, since we make all the optics and the electronics is primarily the only thing, there is a watch battery that's 5 cents. For the current versions of instruments that we make that are designed for just a single person, which is this one, for example, that you look directly, our current estimates of in parts is 75 cents. The closest instruments that we actually do use and compare when we go out in the field is of the order of $2,000 to $3,000. And then the best instruments that you would like, that most of the time the WHO contracts, for example, are of the order of $10,000. And so most common pathologists would not, the instruments that they would buy would be of the order of $2,000 to $10,000. So that's really the difference. But the other aspect of something like this is the reason it's cheap is the fact that we can manufacture. This is primarily designed from a context of scale. So one of the goals that we currently have, those are the number of dimensions that go into making it work perfectly. Because when I said the 10-micron alignment, it's all embedded in that flat sheet of paper to begin with. And the other aspect of cost comes from a perspective of what we would like to do. One of the visions is every kid in the world should have a microscope in their pocket. That's one of the goals. It's an obvious thing personally to me. When you look at the stars, you see sort of starry eyes. The astronomy is so accessible to people. But when you think of a cosmos, a microscopic cosmos is amazing. I mean, last weekend when I had a couple of friends with me and there was a kid, and we were just talking about things like that. I was showing them the microscopes, and he said, let's look at grass. We looked at grass, and I didn't know this. This is sort of what I call a discovery of one. We found, it turns out grass on its edges actually has cells that secrete silica. That's when you pull grass, you cut your hand, right? And then immediately, can anybody guess why would grass evolve to cut you? Yeah? So the tongue. When animals graze, it would cut the tongue as well. And I don't know what the animals do to make sure that they can still graze. But one of the contexts of thinking about something like this is that everyday objects have hidden wonders in which if you were actually carrying instruments around, any single surface, any object that you look at actually has a very interesting structure to it. There are explanations that you really have to wrap your head around. So the connection between global health and education is a simple one. And now I want to turn this more into a business conversation and is connecting the dots. So from a context of sustainable deployment of instruments and things like that, I mean, right now we're doing field trials both in Nigeria and Uganda for 4 different diseases, and we'll decide how things go based on that. The current diseases that we care about is malaria, schistosomiasis, which is after malaria, it's one of the hardest hit from a perspective of economic impact. It's everywhere in China and in India and most of Africa. We're looking at African sleeping sickness, and there are a couple of things that we are looking from a context of filarial diseases. So depending on how the clinical trials go, we're going to try to convince some people. We are going on and manufacturing something like this anyway, but the other aspect of this is connecting the education world with the global health world. So one of the business models that we're going to try is something like the Tom's model of shoes, for example. Yes?
Do the big, like the Zeisses of the world feel threatened?
That's an excellent question. I'm not going to say it on record. No, I think threat is not the right way to think about this. The right way to think about this is their market and the margins that they operate. And the problem that they're trying to solve is very different. This is not thinking that, of course, this is a microscope. But for me, this is a diagnostic instrument. We're making all kinds of things in which some of the staining will be done in the same. They will be disease-specific such that you don't actually mess up with something. If this is the one that you picked up for malaria or schistosomiasis, it has the perfect magnification, the perfect modalities that you need. So it's going away from this idea and saying, what is the minimal that you need to do to really be able to get to the problem you're trying to solve? Zeiss is not in that business. Zeiss is in the business of making research instruments. Yes? That's correct. That's what you saw. That's correct, yeah. And this is actually right there is actually E. coli. Right there is malaria up on the top. So these are all just images from the same instrument. It's a little bit saturated, but there is trypanosome up there. That's Giardia. That's what's traveler's diarrhea. Biggest causes for that is. Yes?
Were those pictures from that?
That is correct. Most of those pictures are actually from this instrument right here. So I want to show you one more thing. I typically do this, but since we are leaving for the field, I want to keep my instruments. But the other thing that I typically show is thinking about robustness. So, from a context of an instrument that you build and design, the current versions that we make have a little plastic coating. You can throw them in water. They actually operate underwater as well. And this is one of the things of thinking about not microscopes that are fragile, that are sitting on an object, because I don't have that image here. The thing that really got me clicking was recently I met somebody in Uganda that works on African sleeping sickness, one of the challenges is if you don't diagnose and treat, the person is going to be dead very soon. So they do screenings. And in one of their screenings, there are these two people who take a 4-wheel drive. They strap up on top of the roof 2 microscopes that are strapped with ropes. They go in there, and they're ready. They enter this rural village and say, okay, line up. We're going to do a screening. Everybody has to give a blood smear. And the heads and the chiefs of these villages line people up. And they say, oh, we forgot to bring a chair. And so then you have this object sitting right in the middle of the road under a tree with people lined up, and that's how screenings are done. So there is that context that really brings this idea of penetration. And it turns out there are basically 5 people for the entire country of Uganda that go out and do these screenings. And so the same surveillance question applies here as well, is the fact that we don't have the infrastructure, both the people and the instruments, to actually penetrate the places we need to penetrate. Okay, I'm going to see if there is any other slide I want to show, but I want to turn this into more of a conversation. This is a 6-year-old that came to one of the events that we did who would not give up until he folded his own microscope. So if there are people like that, we could do it for a few people to just play with, and I could explain a little bit in the more details of how it's actually built. What are the components of an instrument like that? But let's do that after. This is some parts of the machine that we're making that's going to actually do high throughput manufacturing for some of the components. And I want to stop here and just let's turn it into a conversation and we can just talk. And I'll leave this just in case you are interested and want to write an email. Yes? Go ahead.
So what does the folded up one look like? Not the projection one, but the...
Oh, right here. So I can pass these around. For example, the thing that I was showing, this is the non-projection, right? And it has a little switch at the back, just like the other one. But this is a fascinating instrument because this has 9 microscopes built in the same object. And I'm going to now I'll pass this around to people. So this is, just so people didn't see, you slide in, the slide goes in and locks right here. There's a little vernier caliper built in that tells you where you are on the slide, because what we were doing as a search, if I had looked at the vernier caliper, I would know, and then I wouldn't have to be searching. So if I see something and I have a colleague who I want to confirm, you just give it and tell the coordinates. There's a little bit of things built in. For example, here you mark, so you want to do parasitemia count, then you later on the number of dots that you put in an object like this. The way I would use this is that I just bring it close to my eyes and just play with the tension of my hands, and I'm just basically seeing a much larger field of view than what I could show you guys on a projection. But the interesting thing is, some of the optics and the principles are all the same. It's just primarily that the light sources are much brighter. Yeah. Okay. Yes?
Is the outer part made out of paper or cardboard?
So this is a very important paper engineering question because it's a non-trivial question. We actually looked through a large number of stocks of paper, and the reason it's so smooth to actually use is primarily because there's not that much stiction. So paper comes in many categories because paper engineering is a huge field. Our world is littered with paper. There are many, many, many different types based on weight and pounds. So you could call it cardboard or you could call it paper. It's just like a little bit of a stiffer paper. So this is a little bit stiffer than your printing paper, right? Yeah. So it mechanically holds. And why mechanics is important is this instrument, when I push and pull, what you were doing, the optics was moving tens of microns up and down. And that's the hard thing in optics, is to really, the optical component for some of the smallest microscopes are only 200 microns total. So it's like a little grain of sand that you wouldn't actually see, but then you have to move it very precisely. And all the mechanics is built in how it's folded. There's a little linear bearing here that this slides up and down to make sure that the optics moves exactly in sync with the illumination. Yes?
Is this part also paper?
This is all paper. Everything is paper. It's just colored differently.
Okay.
Yeah, and we can pass this through a printer, for example. Yeah, we make many instruments. Some of them are from really thin shim stock like titanium and all kinds of things. There are other versions that we make that are from a little plastic-like paper. So this is another version that we did that we're going to manufacture. So touch that, you feel it, it's waterproof basically. So you could dump this and take it out and carry it in your pocket. Yes?
So last year when I saw your demonstration before, in which he won a wonderful award as a new,
When was that? I don't-
the Baxter Foundation.
I see.
Remember?
Now I know. Yes,
And I
we have
made
met. That's
arrangements
correct.
for you to have
That's
car
correct.
service to the
That
airport.
Actually, so I should put my thank you slide somewhere. That paid some of the money for it. We'll talk about money in 1 second, actually.
to
That's
make
an
your
important.
international flight for your wedding that week.
That is correct.
Yes, and then
Yeah.
you won the award. Anyway, so I mentioned at the time my son is an elementary school teacher in San Francisco. Have you had a chance to connect with the school system and do any of these
So
demonstrations?
that's a very good question. We presented this to a large number of people, and I think we're in a notion of looking for funding. We submitted this to NSF. It didn't go through the first time, and NSF has a big mandate of science education. What we would like to do as a first trial, and so we're writing proposals. This is a proposal that I have written that I've sent out to several foundations, where what we would like to do is, it's very easy for us to make 10,000 instruments. We're going to do as a first trial run, then the next number is going to be 100,000. We're going to give them out, and essentially the only thing we ask from people is to make a discovery of your own self. So any single thing, I run this class at Stanford in which I had all the 100 students build these microscopes. Their only requirement in one single week, they were supposed to ask a question. It doesn't matter whether the world knows the answer or not. I can pluck a hair and ask, how does hair split? And actually, one student asked that question. It turned out it doesn't always split into double. It splits in triplets and fours, all kinds of these crazy things. And so we're basically launching this when we can get the support is it's going to be an online place where people have the same instruments. Whatever the discovery of one, I call it the discovery of one because right now you can go on Google and type in an answer and see what Loa looks like, malaria looks like, and find answer, but it's not your answer. And so the path that you take and you say, I want to find it, cheese. You look at cheese and I found what I found in cheese. So this website is basically going to be a central place all around the world. Anybody can put what they find, and many of the instruments that they will be making and modifying would be exactly the same. This is the other challenge with microscopy. You can go online and find these beautiful images. What you don't realize is a PhD student spent 5 years finding all the antibodies for it to make those glorifying, beautiful images. And I have nothing against beautiful images, but the important aspect of this is to making your own discovery. So the education side is we've given this proposal to the Office of Science and Technology at the White House. They're excited, not in raising money, but in connecting with us to other organizations. We're talking to Smithsonian. There is a large number of organizations. There are a couple of schools in East Palo Alto that we will use as trial, but also we will keep half the section of this trial completely open to people who sign up.
Why didn't you go back to Ghana?
Say that again?
Why didn't you go back to Ghana?
Oh, it's just I spent 3 years there and on and off going back and forth, and we have a little lab there. But Nigeria and Uganda we're primarily going because it has the WHO Microscopy Training Center. And so in the past sites, that was more technology-oriented, but these centers are very essential because this is where all the microscopists pass through. So the current version is really disease-specific. I mean, I think one of the jokes that Jim and I have is we're not going to go everywhere, essentially. We're going to first do some of our own trials, but then make the tools available for people because there are fantastic people in Ghana, Nigeria, and Uganda who both care about infectious diseases and can run these clinical studies themselves as well and decide things for. So I think we don't want to be a bottleneck, and we cannot actually be at most of these places. So we're essentially surveying. There are a couple of places that we are looking in China. India is one thing that we have surveyed, and we'll just find collaborators. Yeah. Yes.
Glass in there, or?
A microscopy talk without using the word lens. That's the first one. So when you think about this, let me just show if I have an image. There are several different types of lenses. If I was to cut this thing in cross-section, this is one image of what you would see. There are optical lenses up there. There are several apertures, and there is a condenser lens, and there is a light source. And it actually changes for the configurations. There are new configurations that we're making in which these lenses will end up being grin lenses. So all those components are first printed on that flat sheet, that the configuration is on flat, and then you put it together. So if I was to literally actually cut this right along this cross-section. The other thing to think about, which is an interesting thing in performance, is in optics you get better by having more number of surfaces to bend light, right? Light only bends when it transforms from one medium to the other. And if you have a gradient index, then it will bend uniformly. So the number of optical surfaces that we put here improves our resolution and the optical performance. And the thing that we are questioning is going disease by disease, because what you saw in Loa Loa is actually pretty good resolution to diagnose that disease. So that instrument will be at that point. There will be other instruments that are even higher magnification that will have more number of these optical surfaces. But still, it will all be micro-optics. And the other area we're actually exploring is better manufacturing techniques for micro-optics as well. And they might have applications at other places. We're interested in this initially. Yes, did I answer your question?
Almost. So you have some plastic things in normal, like everyday terms. You have some paper things and you have
That's
paper,
correct. There's some paper
plastics
things, there are
and-
plastic things, there's a little bit of glass depending on the instrument that you're looking at. The glass is basically a grain of salt, essentially. It's as small. And the materials change depending on the type of instrument that we're trying to make. But at the length scale of the paper, it's all embedded in paper to begin with, or whatever medium we call it. And it's a single sheet. Okay.
Where are you manufacturing these, and what type of manufacturing facilities are you looking for? Talk a little bit.
I think right now we're looking for funding to actually do the large-scale manufacturing. We have a Gates Foundation grant and a Baxter grant that ran out a couple of months ago. So these are 100K grants where we did mostly all the research. And now the next proposal that we're writing is primarily on the idea of manufacturing. Initially, we have different vendors. There are many different directions that we're going to take. Some of them look like spin-offs. It just is going to depend on, we are going to survive, and we will choose paths depending on what comes first. From a context of manufacturing, it's an interesting perspective because of how things scale. So we're doing our own internal trial with 10,000 instruments. And then at that time, it will actually be ready from a context of really going after other manufacturing facilities. Right now, we have individual vendors for different things. And then some of there's another machine that we would like to build that does the folding itself. It takes 3 minutes to fold it. We have other designs that require a single fold that puts everything together. But we don't want to let go of folding, so the kids won't receive a folded instrument. They will receive a sheet. Because the value of this sheet comes from, you got to figure out what goes in this instrument to begin with. And then there will be other sheets that will have all kinds of other components that you can put in to improve the instrument, turn it from a bright field instrument to a fluorescence instrument. So the two manufacturing methods for those will look slightly different. Yes? Yeah?
What scale do you need to reach that price?
That price is for 10,000.
For 10,000.
That's correct. Yeah.
How much does the machine cost that you showed the design of?
Oh, I think that's something that we're making. The machine that we build costs $3,000. But that's really from a context of doing that repeatedly over long periods of time. Yeah. I think whatever we built would be much, much cheaper. This is also from a context of how electronics industry has changed everything. The light source that goes in, it really comes from the MEMS fab and the fact that lighting industry is very excited about turning everything into LEDs. And there are very specific LEDs we use that don't require fancy optics to begin with, so we compensate in many different ways. So it really is a technology curve. We can't take credit for the fact that right now watch batteries cost 5 cents. It's absolutely stunning, yeah. Yes? Did you have a question? Okay.
So aren't there already machines, I have seen
Uh-huh.
demonstrations
Yeah.
that measure out these paper things like for packaging
Yeah.
and cut them?
Oh, yeah, that's what we use. So some of these are exact same processes that are used for making your greeting cards.
Yeah.
It's die cutting. So some of these are just built out of die cutting. In one single shot, you cut 1,000 shapes. Everything that we do, Stanford has a patent, yes. But at the same time, I think the beauty of being at Stanford is we can pursue the inventions that we would like to pursue and take them in directions. For example, if Zeiss, not on camera, but if a big, large optical company wanted to do this, they would have different motivations as compared to somebody who really, one of the things that we talk about often enough is We care about education and we care about global health, but if it was only one and not the other, that would kind of beat the purpose of what we're trying to do. It's very easy to bring something into a US market thinking about the Christmas deadlines, and the fact that it goes out from a Christmas perspective. And a lot of things actually kind of get derailed from that perspective because the big picture is lost. So what we're trying to do is connect those together, and that would require us to play with the pattern. Okay, I think we're already out of time, but I am here. We can keep taking questions. We don't want to hold anybody who has, I am assuming, all kinds of parties that you all have to go to tonight. Yeah.